I'm Dr. Dan Stickler, and I presented this talk at the A4M conference in Las Vegas back in December of 2015. And I had several people contact me and ask me if I could do a recording of the slides. So I put this together, and this is a talk on applying nutritional genomics into your clinical practice. And it really doesn't matter if you are a physician, a chiropractor, a naturopath, a health coach, a nutritional consultant, registered dietitian. This is an appropriate class for you because it takes you through utilizing genomics in the clinical practice to help you develop a nutritional plan for clients. Now, where are we in medicine right now? I mean, we're at a point that we are actually able to transform and transcend the boundaries of evolution and really go into this realm of human potential. We have technology that is merging with human physiology to advance that human potential. We even have pharmacology. I mean, I'm sure many of you are familiar with the movie Limitless, where he took a pill called NZT and he tapped into parts of his brain that were not there before. We also have this new technology with genomics where we have epigenetic modification that can occur, and like in the movie Lucy, where she actually shifts her genomics in order to create a new appearance. We're also experiencing this with CRISPR technology, where we can actually go in and edit genes and change the actual blueprint, the genome. So how modifiable is our genetic code anyway? Well, if you look at some examples just from nature, you have these honeybees, and you have the worker, the queen, and the drone. The interesting component of this is they all have an identical genetic blueprint. There's no difference in those. What the difference is what aspect of those genes is actually turned on and expressed. And the same with the butterfly and the caterpillar. They both have the exact same genetic blueprint, but you can see the phenotypic expression is completely different in both of these forms of this one in particular genotype. Even in the humans, we've developed an epigenetic shift in this person on the left is someone with genetic haplotype that is predominant for equatorial region people. So when she adds additional body fat, it tends to add that body fat around the buttocks area, whereas the girl on the right is more of a Scandinavian haplotype, where she tends to put body fat on all over, and it's just basically the genes that have been turned on and turned off epigenetically over the centuries. So we're talking about a whole new paradigm. We're at a point now, I mean, this whole human genome project has shifted the landscape, and we can now get your individual blueprint through genetics, through the labs associating with those. We can look at the environment in which you, as a genetic organism, and where we have your blueprint, we can look at how you operate and how you do that within that environment. And we can help to write that instruction manual that was essentially missing from human life. We're almost like these aliens that were placed into these bodies and really don't know how to use it. I mean, in my medical practice, we work with people, we teach them how to walk. You look at the teenagers nowadays and even some adults, and their postures, their walking gait, their ability to do a normal human squat, they're all off. And what we do is we teach people how to move within that body and move to create responses. We also teach people how to breathe. We find from a stress standpoint, when we run stress protocols on patients, we do psychophysiologic stress profiles, and we look at physiologic responses to stress. And a lot of the times we find that people just don't breathe correctly. They may breathe 22, 24 times a minute and not see anything wrong with that, when if we get them breathing 10 to 12 times a minute, like is a more normal pattern, then they suddenly have less stress and they're able to function better, they're able to think clearer. So we have these abilities now that we have the blueprints to create this user manual. We can even install upgrades through technology. I mean, there's the cochlear implants, there's bionic limbs that we're using. We can put in lenses in the eye. We can create just about anything now through technology. We can alter our bodies through different fueling mechanisms. We can even, from a brain standpoint, update the operating system. So precision medicine really requires a different type of clinical trial, one that focuses on the individual, not average responses to therapy. So we're going to go into the specifics of the genetics, and we're going to talk about human potential nutrition, and I'm going to take you through our approach with the way we go about this. But before I do, I want to tell you a little bit about my background in nutrition. I went to medical school and had about 2 weeks of normal healthy nutrition. I had 2 full years of pharmacology, but my nutrition was very limited, so most of it was self-taught. Well, I graduated surgical residency in 1999 and decided I wanted to focus on gastric bypass and bariatrics because I really did have a very passionate interest in weight loss and normal human nutrition. But in 2004, I actually discovered the world of age management medicine, where we actually take healthy individuals and we can enhance them through different mechanisms. And I thought, wow, this is really where it is. I mean, step away from the disease model of medicine and into this realm of really creating health in individuals, or helping them to create that health. So I overlapped the two practices for about 5 years, and then in December of 2009, I walked out of the operating room and said, I'm done with it. It doesn't work. I don't believe in weight loss. And that changed in 2010, and I will explain exactly how that has changed. But let's look at some of the training I had in this. Like I said, it was mostly self-taught in the genomics. Thousands of hours of time that I spent cruising the internet, researching articles, reading books. I spent a summer school in nutrigenomics and nutrigenetics in Italy in 2014 through the ISNN. Taken online courses. There's a great course that's offered free through Coursera periodically on epigenetics. It is a course out of the University of Melbourne, and really, I mean, what I learned about genetics in medical school could fill a high school textbook right now. And most of it was not actually accurate anyway. I also use a lot of online resources like OMIM, GeneCard, and Snipedia, but that's how I developed my genetic protocols. Now let's look specifically at where we are from a dietary standpoint in the current scheme. There is not a perfect human diet. We know this. I mean, you can look at this. Look at the U.S. News & World Report, where they ranked, I think it was something around the area of 35 dietary programs, and they ranked them according to what the experts felt like was the healthiest diets. Of course, the top diets: DASH, TLC, the Mayo Clinic diet, the Mediterranean, Weight Watchers, and Flexitarian. You can see a lot of those are pretty divergent groups, yet the most popular, when you look at success and you look at Google popularity, paleo was number 1, and it ranked 34th out of the diets based on the expert evaluation. Atkins was 2, and it was ranked 32nd. Gluten-free, Mediterranean, DASH, and the military diet were the other top-ranked diets. So why is it that you can have these people that are standing up on their stumps and preaching how great these diets are, and yet they don't work for other people, and there's such a wide range of the types of diets we're talking about? Well, the reason for that comes down to genomics. And each person has their own individual genetic code that can change how they respond to different nutrients. So when we take the approach with nutritional recommendations, we take the N=1 diet approach, where we look at the person's goals. What's the goal of your particular plan? Is it health maintenance, weight loss, muscle gain, fat loss, muscle gain together, decreased inflammation, intestinal health? I mean, those are all wonderful goals, and they can all go together. But there's times when we really have to dig down and look at the genes. And now we actually look at the genes first, and we help to guide them in selecting a diet that not only they're comfortable with, but that will work best with their genomic blueprint. So we take real particular look at the genes when we're finding that people have poor weight loss, and this is despite really good compliance with different dietary patterns, and we haven't pinpointed one. Then you want to look at the genetics. If you're unable to get the fasting glucose under 90 despite a very good compliance with the diet, you need to look at the genetics. If you're unable to get the fasting insulin under 5 despite good compliance, you got to look at the genes. If the triglycerides stay above 100 despite a really healthy, low carbohydrate diet, you got to look at the genes and see what's going on. And does total cholesterol go up on saturated fat? It's another time when we really focus in on what's going on in the genomics. So let's look at some of the data that's out there. There's a lot of research right now on the genomic diets, and there are several underway right now, large clinical trials, but these are the two most popular right now. Back in 2014, the European Society of Human Genetics went through a study with approximately 200 overweight individuals, and they had a certain group of people follow a diet that was specifically based on their genetic profile. So whatever their genetic profile suggested, this was the diet they put them on. The other group was randomly assigned, and what they found is the group that followed the diet that was genetically predicted for them, they lost 33% more weight and actually built more lean body mass on that particular diet. Now, there was another study that was done. It was actually initially done out of Stanford. It was the A to Z study, where they worked with a low carbohydrate diet, a low fat diet, a Mediterranean diet, and they were just determining, this was back in 2007, they were determining which dietary pattern worked best, and it was just a randomized dietary plan across all participants. Well, then 2 years ago, a company called Interleukin Genetics went in and they sent out genetic test kits to the participants that were in the A to Z trial, and they looked retrospectively at which ones of those were actually following a diet that their genes predicted they would do best on. When they matched those up, they found that the group of participants that were matched to their genetic diet had 278% greater weight loss than those who were not matched to their genetic diet. That's pretty profound effect there, and it really has opened my eyes to the benefits of a genomic-based diet. Before we get real in-depth into the genes, though, I think it's important that we understand exactly what we're looking at when we look at genetics and use that to determine dietary patterns. So the first thing I want to talk about is what is a SNP. SNP is SNP, and it stands for single nucleotide polymorphism. Now, when we look at a gene has thousands of base pairs in it. Some of those base pairs are what we call introns, which don't really code for anything. And then you have the exons within that gene segment that will code for individual amino acids to create the protein, the receptor, whatever you're creating out of this gene. The exons are the actual coding region. Now, within this gene, both intron and exon, you have what's called single nucleotide polymorphisms. Now, these are changes in base pairs within that gene that actually have been shown to vary within the population and cause some sort of response. So about 1 in every 300 base pairs, there is a single nucleotide polymorphism that can be different based on different haplotypes of different individuals. So we're looking at really 1 in 300 base pairs that can actually have an effect. Now, a lot of these SNPs will actually have zero effect whatsoever, so you've got to know exactly which ones you're looking at and which ones the research is based on, and that's really the understanding of the genomics of nutrition, and that's where it comes into play. So looking at nutrigenetics and nutrigenomics. I think a lot of people get this confused. Nutrigenetics is really a diet that is based on the genes. This is what we're talking about today, is nutrigenetics. Whereas nutrigenomics is where your diet will actually alter the transcriptome and the proteome of the genes. So we're focused really on the SNPs, which is nutrigenetics in this particular situation. So, like I said, SNPs are occurring about in 1 of every 300 base pairs. If you look at a single gene here as an example, COMT, which is catechol-methyltransferase gene, there's 28,000 base pairs there. It codes for a 271 amino acid protein, and within those base pairs, there's 886 SNPs. But 80% of those are in intron regions, so only 20% of the SNPs are actually encoding regions of the gene. Now, sometimes there are promoter regions too that are affected that can affect the gene outcome. But generally, we're looking at the SNPs that are within the exons, and so you're talking about 160 SNPs in the COMT gene. Now, there's only 2 that are really relevant when we look at genomics in our clients, and that's just because those are the ones that we have the data on right now, and there may be variances that occur in these others that have an effect, but as of right now, I don't have the evidence, so I don't use those. Now, it is all based on probability, so we don't know which of these genes are actually turned on or turned off. All we know is that probability-wise, if you carry this particular single nucleotide polymorphism, you're going to have this outcome. So let's look at what I use when I'm looking at a person's nutritional genomics. This is an older template that I had for Livewello. So you can go into Livewello and you can actually create your own genetic templates in there that can extract the data from programs like 23andMe, and that's what I use in my clinical practices. I have my clients get 23andMe genetic testing, and on the back side of that, they actually code 635,000 SNPs. And you can download that raw data file and then upload it into Livewello, where you can extract the particular ones you want to see. And when you do that, you can actually look at this one page and you can have all these SNPs that you can work with right here. So just as an example, right now, some of the SNPs that I look at for dietary fat, and I'm going to go through a couple of these. I don't have time in this lecture to go through all of them. And we generally do clinician training where we go through all of the SNPs. We go through about 250 different SNPs that can guide clinicians in developing a health and wellness plan for their clients. But the major one, and the one you hear about most, is PPAR gamma. This activates fat cells to differentiate into a smaller size. And a high-fat diet induces adipocytes to transform from small to large, which they secrete insulin resistance factors such as tumor necrosis factor alpha or resistin and free fatty acids. So it's not really a good thing to have happen. In the CG variant, so if we look at the SNP that the person has for PPAR gamma, if they're a CG, it's the one that tells you that the person should probably avoid high-fat diets. Now, if they have a CC genotype, then they have what's called the genetic privilege, where they can work really well with high-fat diets or low-fat diets. And the GG can generally go either way. If you look back at this slide, you can see the PPAR gamma down towards the bottom is a CC, and you can see the phenotype is negative on that, whereas the risk gene is G. And this basically means this is a person who has genetic privilege when it comes to dietary fat. But this is only one gene to look at, but it's a very important one. So this is going to be one of the first genes I look at when I'm determining what the best dietary pattern is. And there is a kind of this layout, which I used initially as an overview, is based on one of the studies on gene-based dieting, and based on the PPAR gamma and ADRB2 genetic SNP, and you can see the RSID codes there for them. And if you go down through this, you can get an idea of what the best style of diet is. This is a very elementary approach at this stage because we know so much more than what this is telling us. But this is just an example of how the genomics can be potentially used to determine dietary patterns for people. Now, there are other genes like insulin resistance genes, and I'm going to give you an example of a client that I had. She is a 50-year-old female, and she's been with me for 2 years, and this was really during the time that I was developing my genetic program. And she'd come to me on paleo, and she had not been able to lose weight, and she's very active. I mean, she's only 10, 15 pounds overweight, but very, very active. 4-0 tennis player and a meticulous record keeper and a biohacker. I mean, she tested everything. We had her on the paleo diet. We tried some calorie restriction without any success. She even wanted to try full ketogenic, so she went full ketogenic. She measured ketones. She stayed in ketosis and still could not lose any weight. And in fact, her fasting insulin levels always stayed in the upper 90s. And her fasting glucose levels and her fasting insulin levels always stayed around 7 or 8. And we couldn't figure out how this was happening despite such good compliance with almost no carb intake whatsoever. Well, it was about that time that I started looking at insulin resistance genes, and I ran her insulin resistance genes, and sure enough, she was full of them. Her problem was in her genomics. Now, this isn't something that can very easily be taken care of with just dietary patterns alone. What I focused on with her was to work with pharmacogenomics. So we put her on metformin, which is also known as glucophage. It is a first-line diabetic drug, but it's not just for diabetics. It's really good about activating AMPK in the cell. And when you activate that, you actually get increased insulin sensitivity, you get suppression of mTOR, you get alterations in gut bacteria. So metformin is a really awesome treatment. It's also a great anti-aging treatment, by the way. There's a study going on in New York right now called the TAME study, Targeting Aging with Metformin. Really good study. Now, if you can't use prescription medication to treat this, you can also use an herbal called berberine, and berberine will actually activate AMPK as well through a little different mechanism, but it creates a similar response, albeit not as extreme as really you get with the metformin. So let's look at a couple other genes that are relevant to us. Tumor necrosis factor, this one is RS 1800-629, and the risk allele is A. It is a very difficult one to work with when A is present. And I found this in multiple clients. They have a great deal of inflammation. They tend to have high risk for a lot of chronic diseases. And with this, if you can't get the inflammation down, you're going to have a lot of trouble getting success with dietary patterns, creating the response that you want to see. So we really focus on getting inflammation down first and foremost in this group. Another interesting genetic SNP is the vitamin D receptor Fok. This one, and you can see the RSID, this one codes for the vitamin D receptor, and we look at actually quite a few of the vitamin D receptor SNPs, but this one plays an interesting role in insulin resistance. And if you are if you have an at-risk allele, and you supplement with vitamin D, you will get greater insulin sensitivity. If you have the wild type, if you have the normal alleles, then you generally don't have the propensity for the insulin resistance. But if you do have the risk factor, you can add vitamin D and actually increase the insulin sensitivity in these people. Gckr is another really cool gene to look at. It regulates glycolysis in the liver. These people tend to have really high triglycerides and increased CRP. But the interesting thing is when you get their fasting blood sugars, they tend to be fairly normal. But you look at their 2-hour oral glucose tolerance test, and they are really high on this. They seem to have not only problems with glycolysis in the liver, but they also have decreased beta cell function. So these are ones that when you're working with them, you really want to focus on a lower carbohydrate diet in order to get success with this. We also look at a lot of appetite genes. I'm going to go through some of these, and these are only a few of the appetite genes we look at, but you can see Some of these genes like COMT, MAOA, DRD2, and AMKK1 really code for dopamine levels in the brain, and those all go along with the pleasure response, but they do have a profound effect on appetite as well. FTO is the fat gene. This was one of the first genes identified relating to obesity, and that's why it was identified as the fat gene. When people have the variants of this particular genetic SNP, tend to have increased hunger, increased impulse eating, decreased post-meal satiety, and if they are homozygous for the risk alleles, so they are the ones that are most prone to all of these, they also have a preference for meat and salt. When you have normal or wild type alleles for this, you tend to have less of a desire for the meat and salt in general in the person who carries it. We look at genes for metabolism too, so the NERG1, the leptin receptor gene, LEPR. And this one's a big one because people with abnormalities of the leptin receptor genetic SNPs tend to have real problems with metabolism. PPAR GC1 is an interesting one when it comes to metabolism as well because it's a regulator of mitochondrial biogenesis and function. And it really tells you whether endurance training is going to be most beneficial for fat loss in somebody. And it can determine whether they get any kind of a response to cold thermogenesis, where the white fat is really converted into more brown fat. It's also involved in mitochondrial protection, so it works with NRF2 and SIRT1. You really don't want to have too many abnormalities with the PPAR-GC1 genetic SNPs because it really is a big deal when it comes to oxidative stress, obesity, insulin resistance, all the really major issues that we want to avoid. We can look at certain genetic SNPs that have to do with carbohydrate intake. So all of these have to do in particular with some aspect of carbohydrate intake. It could have to do with taste, it could have to do with the utilization of the carbs, it could have to do with abdominal fat. And we have one in particular that tells us whether we will do better on a higher protein diet or a lower protein diet. So looking at some of the carbohydrate genes, the PLIN1 is one I think that people find really interesting. This really is the carb gene, and if you carry the T variant here, this is somebody who needs to take starchy carbs in order to decrease abdominal fat and create fat loss in the gut. And I've had a lot of people that were eating Atkins or paleo style and really minimizing any carbohydrate intake and could not lose the abdominal fat. We found that they had the PLIN1 genetic variant, and we had them add starchy carbs, and sure enough, they started losing the abdominal fat, which was really the problem to begin with, is they were not taking enough of it. Another gene set that we look at has to do with taste, and two of these are really big deals. The TAS1R3, it really has to do with how we perceive the sweet taste. Is it strong? Is it weak? So if you have the risk allele, you have a diminished ability to taste the sweetness of sucrose, and therefore you tend to take higher amounts of it to satiate any sweet cravings that you might have because you just don't get that full sweet taste with it. And then there's the TAS2R38. Now this is one that I can tell you a lot of people have this. This is it really has to do with the ability to taste bitter. You know, the little test you did in grade school where you said, do you taste this bitterness or not? Coffee and broccoli tend to taste more bitter to it. But the interesting aspect is that these people can also become super tasters. Now, you know these people. It's the person who, if you have the cookie jar filled with cookies and you walk by and you take a cookie, you find yourself keep going back until the cookies are gone. This is really more pronounced in the females that carry the AA genotype than it is for the males, but it's pretty classic, and the ability to predict this is over 95% in my experience with my clients, and I've done over 1,000 genetic evaluations for nutritional habits, and this TAS2R38 is a really strong one, And what I tend to do is have people avoid situations where they will have an unlimited supply of the sweet that's driving them to go for it. So as long as they have the ability to not have access to unending amounts of this stuff, of these sweet things, I tell them that it's okay to take it. The ABCG8 gene is a gene that we call the anti-vegetarian gene because it has to do with plant sterols and the amount that you end up absorbing from the gut. Now, this is a big deal, and most people don't understand that, but plant sterols have a significant association with heart disease when they get too high. And variances of these particular genetic SNPs can cause you to absorb a great deal of plant sterols. So people who eat a more plant-based diet that carry this are going to have very high triglycerides and high cholesterol and actually have a very high cardiovascular disease risk propensity. So you look at whether they have it or not, you look at their post-meal glucose, and you really want to focus on their cholesterol. So do they have high triglycerides based on eating a high plant-based food diet? And in that situation, you want to really guide them into eating less of a plant sterol-based diet. ApoE is a big one for many people. In fact, in 23andMe, you have to check off that you're comfortable going ahead and looking at the results of the genetics that you have for this, because there are major health implications. I mean, you're talking about 12 times the standard risk for Alzheimer's disease if you have ApoE4 genotype. But you need to have both genetic SNPs coded in order to find out what exact genotype you have, as you can see from this chart here. And the risks are pretty huge. I mean, you're talking about 11 times the standard risk for early onset Alzheimer's, and for late onset Alzheimer's, it's almost 60 times the standard risk. And that risk goes up even more if you consume alcohol or if you consume a high-fat diet. But these are really important genomics to know in each individual, and I think almost everybody should have this to understand where they are. Vitamin D is a really complicated one when it comes to genomics because you've got to look at multiple different components. The CYP27B1 actually converts 25-hydroxyvitamin D, which is what we typically measure when we're measuring vitamin D levels in a person, into the active form of the 125-hydroxy. So if they have a genetic variant here, they don't convert the 25-hydroxy into 125 very well. So you may be measuring 25-hydroxy and say, hey, they got great levels. You know, they're 40 to 60 on their vitamin D level, but you're looking at 25-hydroxy. If they're not converting that, then they are going to have higher levels because it's going to backlog a little bit. And then if you actually check the 125, you may find that they're deficient. So you need to keep their 25-hydroxy levels in the 80 to 100 range in order to get normal levels of 125. But what I do is if I see this, I tend to order the 25-hydroxy as well as the 125-dihydroxy vitamin D. Now, the other end of this is the CYP24A1, which takes the active form, the 125, and converts it into the less active form. So you may have a low level of 25-hydroxy, which is what you're measuring in the blood, and they have a normal 27B1, so they probably have elevated levels of 125 because they have variances of this CYP24A1, which impairs their ability to break that down. Now, Clotho is a really cool one too because Clotho actually inhibits the 27B1. The reason I mention this one in particular, though, is this is one of our strongest longevity genes. You carry the GT variant of Clotho and your longevity is amazing. This is one of the ones that everybody wants to have when I check their genetics. Now, I talked about the vitamin D receptor earlier, and I talked about the VDR-FOK, which we use to help create insulin sensitivity in someone with insulin resistance that has a variant of this. But there are also low responders to vitamin D. That's why supplementing with vitamin D tends to help regulate this a little bit better. Now, the VDR-BSM has an upregulation of 25 to 125. So this works very similar to the 27B1 that we talked about earlier. So the person may appear low and in need of more supplementation, but this could lead to toxicity. VDR-TAC, when it comes to the vitamin D receptor here, individuals with the T variant, which is the risk variant, low sunlight conversion of vitamin D. And you also see a higher incidence of MS and seasonal affective disorder in this group. So in closing this out, I just want to say, as clinicians, we really need to focus on learning the genomics. There's a lot of direct-to-consumer testing out there, and they're great, but you can go to 3 different labs and get 3 different results because it's all based on which particular genes that they use to determine what the best dietary recommendation is for somebody. We can look at other genes that have to do with supplementation. Does somebody need more vitamin A? Do they need higher doses of vitamin C? Do they need to supplement with choline? Should they avoid vitamin E because it causes inflammation? A lot of this we can determine from the genomics, and so I think it's imperative as clinicians that are doing any counseling in nutrition, they learn this genomic basis of human nutrition and become the expert. Be the person who not only can get the testing done, but can actually interpret the testing. It's just kind of like using labs from a blood test and not understanding what they mean, but just telling somebody they're abnormal or normal based on the normative values. It just doesn't work very well. You want to be the person who can actually read this like a foreign language and translate it for the client. So lead this paradigm and become the Iron Man that you're really capable of when it comes to really getting into the new human potential paradigm. Now you can find more on me at the Apeiron Center for Human Potential. That's apeironcenter.com. That's spelled Apeiron. You can also listen to my podcast, The Iron Man Executive, where I talk to a lot of movers and shakers in the genomic world as well as the world of human potential optimization. Or you can email me directly if you have any questions at drstickler@apeironcenter.com.